Hello and welcome to some online class. Friends, today we shall discuss about the splicing of pre-mRNA. Pre-mRNA or the precursor mRNA is the primary transcript. This primary transcript undergo processing to form mature functional mRNA. During processing, the non-coding sequences known as introns are spliced out and the exons are joined accurately to form a properly functional mRNA molecule. Like the splicing of tRNA precursor as in case of yeast and the rRNA precursor as in case of tetrahymena thermophila, the splicing of pre-mRNA also occur in two different stages, but there are certain differences among them. First difference is that the splicing of pre-mRNA is not autocatalytic. Second difference is during splicing of pre-mRNA, there is no involvement of splicing nuclease and splicing ligase. The process of splicing of pre-mRNA is carried out with the help of RNA protein complex known as spliceosomes. So spliceosomes are the complex RNA protein structure that carry out the process of splicing of pre-mRNA. Spliceosomes are like small ribosomes. They contain a set of small nuclear RNA and about 40 different proteins. Spliceosomes are the ribonucleoprotein structure. They are composed of different small nuclear RNA and different types of proteins. There are 5 small nuclear RNA which participate during splicing as the component of the spliceosome. The spliceosome contain 5 types of small nuclear RNAs. The small nuclear RNAs are small nuclear RNA U1, U2, U4, U5 and U6. Another small nuclear RNA, that is small nuclear RNA U3, it is localized in nucleolus, where it is probably involved in the formation of ribosomes. So these 5 small nuclear RNAs are the components of the complex RNA protein structure known as spliceosome. These 5 different small nuclear RNAs, they form specific RNA protein particles known as small nuclear ribonucleoprotein particles. So these 5 different small nuclear RNA form 4 types of small nuclear ribonucleoprotein. Small nuclear RNA U1 form a complex particle known as snRNP-U1. That means small nuclear ribonucleoprotein U1. Similarly, U2 snRNA, it form another snRNP, that is known as snRNP-U2. Another snRNA U5, it form another snRNP, that is known as snRNP-U5. And U2 and U6 snRNA together, they make another snRNP, that is known as U4 U6 snRNP. So totally there are 4 different types of small nuclear ribonucleoprotein which are assembled to form the spliceosome. So the spliceosomes are assembled from 4 different types of snRNP. Each snRNP is composed of a subset of 7 well-characterized snRNP proteins plus 1 or more proteins unique to this snRNP. So this is about the various components of spliceosome. So spliceosome is a complex RNA protein structure that carry out the process of splicing of pre-mRNA. The process of splicing of pre-mRNA can be divided into 4 different steps. Friends, this is the primary transcript or the pre-mRNA. This pre-mRNA contains 2 exons, exon 1 and exon 2. In between there is an intron. And there are 2 different splice sites. This is the 5' splice site and this is the 3' splice site. And there are 2 dinucleotide sequence, that is GUAG. These 2 dinucleotide sequences are the completely conserved sequences. In the first step, snRNP-U1 bind to the 5' splice site. This is the 5' splice site. So first of all, snRNP-U1 bind to the 5' splice site. After binding of snRNP-U1 to the 5' splice site, another snRNP, that is snRNP-U2, bind to the branch site. This is the branch site which contain a adenine residue. In step 2, the complete spliceosome assembled. This complete spliceosome contains 4 different snRNP. One is snRNP-U2, snRNP-U5, snRNP-U1, another snRNP that is composed of U4 snRNA and U6 snRNA. After assembly of the complete spliceosome, the spliceosome cleave the transcript at the 5' splice site. This is the 5' splice site. This 5' splice site has been cleaved by the spliceosome. Next is step 3. In step 3, the 5' end of the intron is joined to the A in the branch site to form a lariat structure. So friend, this is the 5' end of the intron. This 5' end of the intron has been joined to the A residue in the branch site. This is the branch site, and in this branch site there is a adenine residue. So this 5' end of the intron, it binds with the A residue that is present in the branch site to form a lariat-like structure. After the formation of lariat-like structure, U1 and U4 are released. Next is step 4. In the step 4, the 3' splice site is cleaved. This is the 3' splice site. So in the step 4, this 3' splice site is cleaved and the 5' end of the exon 2. This is the exon 2. The 5' end of the exon 2 is joined with the 3' end of the exon 1. Thereby, these 2 exons are joined. And this is the mature mRNA. And this is the lariat-like structure. So friends, this is the spliced mRNA. So due to the joining of exon 2 and exon 1, the final product is formed. And the final product is the mature mRNA, which is then translated to form a polypeptide chain. And this is the lariat-like structure that is associated with these 3 snRNP. After that, these particles also dissociate. So friends, this is about the splicing of pre-mRNA. For more updates, please subscribe my YouTube channel Sam Online Class. Thank you very much.